Hello, everyone, and welcome to another exciting talk in our GPCR webinar series. We'll get started now. Today's webinar is titled Mu-opioid Receptor Splicing and Signaling, Another Dimension of GPCR Function, presented by Professor Pasternak, who is the Head of Molecular Pharmacology and Chemistry Program at Memorial Sloan Kettering Cancer Center. On behalf of DiscoverX, I would like to thank you all for attending this webinar. I'm your moderator, Alpana Prasad, Product Manager for GPCR Assays at DiscoverX. Before we begin our presentation, I would like to remind you all that this webinar is designed to be interactive, and you can submit your questions by typing them in the Q&A box on your screen. All questions will be answered in the Q&A session at the end of the talk. The presentation slides and the recording of today's webinar will be available on the DiscoverX website, and the link will be emailed to you tomorrow. Dr. Pasternak, if you could move the slide forward. Before we get started with the presentation, I would like to take a couple of minutes to give a quick overview of DiscoverX's solutions for drug discovery and development from target ID to post-launch for some of the folks in the audience who may be new to DiscoverX. DiscoverX is the leader in cell-based assays. These assays are available as both off-the-shelf products and as a part of our screening and profiling services. The portfolio covers several druggable target classes, including GPCRs, kinases, interleukins, checkpoint receptors, etc. And you can choose your target from over 900 pharmacologically relevant functional assays. With over 15 years of experience and over 1,700 publications across multiple applications, these solutions deliver reliable data to support and drive any drug discovery program. Next slide, please. As you know, GPCRs represent the largest class of validated therapeutic targets, with over 30% of currently marketed drugs targeting these receptors. DiscoverX offers the largest GPCR assays portfolio that includes over 650 functional assays, covering more than 90% of this receptor class. The assays are available with cyclic AMP, calcium, beta-arrestin, and internalization as functional readouts that can be run in a single or multiplex mode. These naturally coupled receptor cell lines have been tested and validated with their known ligands to ensure superior performance right out of the vial. These assays are available as stable clonal cell lines or complete ready-to-use kits optimized for small molecules or biologics. These assays are also offered as a part of GPCR screening and profiling services. You can also create your own assays in any dividing mammalian cells using our toolbox products. These include vectors, retroviruses, and parental cells with beta-arrestin internalization and trafficking as functional readouts to analyze GPCR variants, orthologs, etc. This toolbox approach was used to develop the beta-arrestin recruitment functional assay to study the E4M splice variants that Dr. Pasternak will discuss today. With that brief overview, I would like to introduce our today's speaker. Next slide, please. We are very excited to host Dr. Pasternak as the speaker today. Dr. Pasternak holds the Ann Barrett Tandy Chair in Neurology at Memorial Sloan Kettering Cancer Center and is also the laboratory head for the Molecular Pharmacology and Chemistry program. He is also the Professor of Neurology and Neurosciences at the Weill Cornell Medical College. Dr. Pasternak's research has focused on opioid receptors and their mechanisms of action, and developing a pain medication derived from opiates that will be as effective in managing pain without the side effects. He was awarded the 2013 Harrington Scholar for Innovation and has been the recipient of several distinguished awards. He has published over 400 articles and been granted 3 patents with several pending. He serves on the editorial board of 8 international journals and also as scientific advisor at several pharmaceutical companies. Dr. Pasternak holds MD and PhD degrees from the Johns Hopkins University and completed his clinical training in neurology at Johns Hopkins Hospital. In today's webinar, Dr. Pasternak will describe a set of mutant mouse models that his lab developed to investigate the importance of OPRM1, 5' and 3' splicing, and the role of their distal C-terminal tails in mediating complex morphine actions. These models revealed major behavioral and transduction differences, including biased signaling and β-arrestin recruitment among the 3' splice variants and the drugs tested. So without further ado, I would now like to hand you over to Dr. Pasternak. Dr. Pasternak?
Thank you. Today's talk will focus on the opiate receptor as a model for understanding the role of alternative splicing within the GPCR family, something that is actually fairly common. What we'll do is discuss the new opiate receptors with a bit of an overview, and then go into the splicing of the OPRM1 receptor, and then describe some of the reasons implicating these splice variants in terms of functional roles, and then finally go into some of the more biochemical aspects of the splicing in terms of bias signaling and beta-arrestin recruitment and GDP gamma S stimulation. Just as a very quick overview, opiate receptors are members of the GPCR family. This family is traditionally thought of as 7 transmembrane domain receptor family of proteins with an extracellular amino terminus and an intracellular carboxy terminus. As you've heard, there's quite a few of these GPCRs in the world, and approximately half of them are associated with taste and smell, and the other half have provided major sources of targets for drug development. To give you some idea of the extent of alternative splicing, perhaps in an area other than the opioids, I took this paper from Einstein et al. From 2008, where they looked at smooth muscle cells in airways and did a random search for all GPCRs, and they found 353 expressed in these cells. Of those, approximately 192 had evidence of alternatively spliced isoforms, with each having an average of 5 different forms that were made. Thus, as you can see, the incidence of alternative splicing within the GPCR family is actually quite extensive, and this has to be taken into consideration when developing new drugs and trying to understand GPCR function. In that paper, they conclude that alternative splicing is a major mechanism that diversifies the GPCR family, and what I try to do today is give you some indications and examples of how this can be supported within the opioid field. The opioids are among the oldest of drugs. They've been around for literally thousands of years. Thousands of derivatives have been synthesized, and indeed they were synthesized long before the receptor was actually identified. There's extensive clinical experience with these drugs, and from this clinical experience, several observations came forward. First, although most of the drugs that are made act through mu receptors, it's clear that they don't work equally well in every person. Cross-tolerance among the drugs is often incomplete. Side effects can vary from one drug to another within a single patient. And pulled together, these clinical observations imply that there may indeed be subtypes of mu receptors that were actually suggested prior to their identification at the molecular level. Experimentally, the opioid system has many advantages. The availability of a wide range of ligands is perhaps one of the greatest, but also the ability to correlate molecular binding and molecular function with behavioral actions in a relatively simple and straightforward manner. There are many physiological actions that are mediated directly by opioids. Here's a crystal structure of the mouse mu-opioid receptor as published by Manglik et al. Several years ago. As you can see, the ligand they have here, beta-phenyltrexamine, is seated within the binding pocket, relatively near the outside of the cell. It has, as you can see here, most interactions with TMs 3, 5, 6, and 7. Also note that TM1 is relatively removed from the binding sites for the ligand. Described another way, here's an amino acid sequence of the receptor, and I'm presenting this to give you some idea of how the protein is organized. If you take a look, the receptor is traditional in the sense that it's got an extracellular N-terminus and an intracellular C-terminus. It undergoes extensive splicing, and in this particular receptor, there are 4 exons that are important in actions. Exon 1 encodes the extracellular N-terminus in the first transmembrane domain. Exon 2 takes the next 3 transmembrane domains. Exon 3, the last 3. And as you can see down in the lower corner here, exon 4 encodes only 12 amino acids of the whole sequence of the receptor. And indeed, even within the C-terminus, these are a very small fraction of the sequence that is actually in that terminus. And yet, what I'll show you later is that substitutions at this position can greatly impact the function. There are many reasons why opiates can differ. One is pharmacokinetics. The other is metabolic profiles, and this has been clearly shown with codeine, where fast and slow metabolizers have very different potencies and effectiveness. But recently, there's a lot of evidence to suggest that once bound to the receptor, differential functional activation, or bias signaling, can play a major role, particularly in the opioid receptor system. On top of that, there's been the identification of multiple new targets, each one of which is susceptible to bias signaling. So this presents a great complexity in terms of understanding opiate action. This provides a schematic of the general splicing patterns of the mu-opioid receptors. These splicing patterns are maintained in mice, rats, and humans, and there are 3 major predicted forms of the receptor. The traditional 7-transmembrane domain receptor that you can see here is the classical GPCR. As you can see, the gene has an exon 1 promoter, which starts transcription right before exon 1 and generates the 7 transmembrane domain receptors. There are others that it translates as well, a single transmembrane receptor that have basically skipping of exon 2 that you can see here, and that leads to frame shifts and only a single transmembrane domain. We won't talk about those today. There's a second promoter upstream from exon 1 that's associated with exon 11. And as you can see here, the exon 11 promoter is able to activate the entire gene. The exon 11 promoter has a TATA box, unlike the exon 1 promoter, and it appears to be under differential control. In humans, there are 12 different identified 7-TM splice variants. There are 3 variants containing this truncated 6-transmembrane domain structure. That results from the skipping of exon 1. So it goes from exon 11 directly to exon 2. So since exon 1 had that first transmembrane domain, that is now missing. And there are 4 variants that have that single transmembrane domain that we talked about just a minute ago. This schematic gives you perhaps a better insight on a detailed level. And what it shows is that for the 7-TMs, the full-length variants, they share the same exons 1, 2, and 3. Now, this is important because these are the exons that comprise all the transmembrane domains. And indeed, these are the sequences that were shown in the crystal structure of the mu receptor. To be able to make crystals, the laboratories that have been doing the studies have truncated both the C-terminus and the N-terminus. So what we see with the crystal structures is really the central portion of the receptor without those 2 ends sitting there. With the 7-TMs, what you can see is that exon 4, which has 12 amino acids, as you can see over here, is replaced by various different combinations of exons, each of which has a totally separate and different amino acid sequence. It's also quite notable that they vary enormously in terms of their length. So, for example, MOR1B5 only has 2 amino acids, whereas MOR1F has 58. You can see a similar pattern of 3' splicing within the 6-TM variants, but notice that these don't have exon 1 and therefore lack the first transmembrane domain. There's been much evidence to suggest that these are relevant. Much of it is indirect. First, the conserved structures in mice, rats, and humans, the distinct regional distributions within the brain, the cell-specific splicing patterns, which you can see on the bottom here. Looking at the dorsal horn, you can see the exon 4 epitope labeled in green is located very nicely in the dorsal horn, exactly where you might expect to see it for a mu receptor involved in pain modulation. Similarly, if you use an epitope to pick up exon 7, 8, 9, it too is located in the dorsal horn. However, when you look at high power using confocal microscopy, what you can see is that they are not expressed in the same cells. As you can see, the red dots and the green dots are basically separate. We see no evidence of yellow. They have different pharmacological profiles, which I'll show you in some of the models that I'll present later. And they have differential modulation of morphine exposure. They also have differences in terms of their association with other molecules, specifically CGRP, calcitonin gene-related peptide. As you can see in the bottom right, you can see that the exon 7, 8, 9 epitope co-localizes with CGRP. I can tell you that, although not shown in this particular slide, the exon 4 epitope does not. Furthermore, the exon 7, 8, 9 epitope is located almost exclusively presynaptically, whereas the exon 4 epitope at the level of EM is both pre- and postsynaptic. Thus, all these indirect measures suggest that indeed these different receptor splice variants are truly relevant. If you take a look at the abundance of these receptors, several things become very striking. If you take a look at MOR1, you can see that it is without question, when looking at the whole brain, the predominant isoform. And in fact, if you look at all the others, they're actually, the actual abundance is actually relatively low. And many people felt that this was a little concerning and suggested that perhaps these were not particularly relevant. However, a very different picture comes out when you actually do a regional distribution as opposed to looking at whole brain. Perhaps some of the more dramatic differences would be, for example, in the prefrontal cortex, where in this particular situation, MOR1 itself is now a very minor component, and MOR10, a 7-TM with the exon 7 in the place of exon 4, represents close to 40% of the full-length MOR1 splice variants. That contrasts with the striatum, which again has the vast predominance of MOR1 as opposed to the others. And if you take this all the way across, you can see various different patterns for the different receptors, implying region-specific and cell-specific splicing. These different subtypes are also present in humans. In samples from human prefrontal cortex, we were able to observe a varying abundance of a number of these different receptors, both 7-TM, 6-TM, and single-TM. These receptors are also modulated by morphine exposure. In studies in which mice were administered morphine for a total of 6 weeks, we can see very dramatic changes in the abundance of these different receptors. Again, if you look at whole brain, you see very little change. You see 3-fold or a 5-fold increase in the overall expression levels. This changes dramatically if you take a look at individual regions. So, for example, MOR1D, which is present, and as I'll show you right here, MOR1D has a very low abundance in whole brain. With morphine exposure, its levels in the striatum are increased by 150-fold. It's 300 times more abundant in the hypothalamus after morphine exposure. You see similar patterns with many of the other splice variants, including the 6TMs, where you have a 90-fold upregulation of MOR1G in the hippocampus, a 24-fold increase of MOR1N, and MOR1K, which has been implicated by several groups in opioid hyperalgesia, has a 40-fold increase in the hypothalamus with morphine exposure. So clearly, when one looks at different regions, you can see very dramatic changes in mRNA expression that vary from region to region and vary from splice variant to splice variant. How might you be able to actually explore the functional role of these three sets of splice variants in vivo? Years ago, an exon 1 knockout mouse was generated by John Pintar, and this mouse was very interesting from several perspectives. First, it clearly showed that the mu receptor that was cloned was important for morphine actions, indeed replicating results from other people with their own knockout models. But Dr. Pintar's model was unique in several ways. Most importantly, in his model, the expression of the 6 transmembrane domain receptors was retained. Now, I should point out that not all mouse models are the same. They have different sizes and locations of their disruptions. Even those that are targeting exon 1 may not necessarily lead to the same type of knockout. So it's very possible that any knockout on any exon can lead to a total loss of all the receptors from that gene. More uncommonly, one might see a selective loss of a subpopulation of splice variants by a single exon knockout. And indeed, this is what we've seen with the Pintar mouse. To my knowledge, no one's ever checked the other exon 1 knockout models to see for continued expression of the 6TM receptors. The knockout models that target exons 2 or both 2 and 3, as you can see here, are going to eliminate both the exon 7, both the 7TMs and the 6TM receptors, giving you almost a full null knockout. We made an exon 11 knockout, and in this knockout, we were able to retain full expression of the 7TM receptors, losing only the 6TM receptors. We've also made another knockout, which I'll describe in a little later, in which we knocked out both exon 1 and exon 11. And as you can imagine, by doing that, we have a full null knockout with no expression of any OPRM1 splice variants. There's another animal that's been used, and that's referred to as the triple knockout, and basically this comes from the Pintar laboratory, where he took his exon 1 knockout, as you see over here, and he crossed it with a delta and a kappa knockout. So basically he's lost and removed all the traditional opiate receptors, the 7TMUs, the deltas, and the kappas, leaving only the 6TM opioid splice variants being expressed. Now, if you compare the different receptors with regards to morphine actions, you see a couple very interesting things. In the Pintar mice, if you knock out exon 1, morphine, in terms of analgesia, is totally inactive. However, in the exon 11 knockout mouse, morphine retains full activity, and the potency is minimally changed, suggesting that the exon 11 variants are not important whatsoever for morphine actions, whereas the 7TMs are. Respiratory depression, as you can see in the exon 11 knockout, was unaffected, indicating that respiratory depression is mediated through the 7 transmembrane domain variants, the traditional ones. But what was intriguing was when we looked for hyperalgesia in the bottom panel, and here you can see the saline on the left in the wild type, and then morphine treatment, looking at baseline latencies 24 hours after the dose of morphine. You can see a hypersensitivity to morphine in these animals. This is something that's been described by many groups over the years, and if you replicate the morphine exposure and testing the next day, you can see that it persists for 4 days. If you do the exact same experiment in the exon 11 knockout, what you see is the loss of this hyperalgesia. A summary is provided in the table on the left, indicating that most of morphine actions that we are associated with, that we associate with the drug, are mediated through the traditional 7-TM receptors. However, there's a suggestion that tolerance, hyperalgesia, hyperlocomotion are at least partially mediated through exon 11 receptors. If you actually take a look at other drugs in the exon 11 knockout, what you can see is that morphine and methadone retained full analgesic activity with no significant shift in their potency. But what was intriguing to us, in addition to the compound that we synthesized, IBNTXA, which we've used extensively to study the 6-TMs, if you take a look at other compounds like fentanyl, buprenorphine, levorphanol, and butorphanol, these are drugs that are used clinically. But they also have an important role for the 6-TMs in their actions as well. Indeed, with buprenorphine, if you take away the 6-TMs, buprenorphine is totally inactive and has no analgesic actions whatsoever. Looking at the IBNTXA in a little more detail, what you can see here is an opposite pattern that you saw with morphine. That in the exon 1 knockout, in fact, this is the triple knockout, so indeed there are no delta or kappa receptors here as well, you maintain full analgesic activity. In the exon 11 knockout, where morphine is fully active, you lose all IBNTXA actions. Unlike morphine, however, which, as you can see here, produces a nice respiratory depression in terms of respiratory rate, you can see that the same equianalgesic dose of IBNTXA was without effect. Similarly, if you chronically treat mice with morphine, you get an established withdrawal profile. There's no evidence of physical dependence with chronic IBNTXA. And finally, in this slide, what you see here is a condition-place preference, where morphine shows significant condition-place preference, whereas IBNTXA shows neither reward nor aversion in this model. Finally, to conclude that the role of the 6-TMs, we felt it was important to do a gain-of-function study. Here we use that exon 1, exon 11 double knockout, where we lost exon 11 and exon 1. As you can see, PCR shows that with these different primer locations, the complete absence of any of the splice variants of the OPRM1. So this animal is a complete OPRM1 null knockout. What we did here was we took a lentivirus and we administered the lentivirus to these knockout mice, and the lentivirus that we designed expressed a 6-TM and only a 6-TM splice variant. As you can see, reconstitution of the 6-TM splice variant here in blue showed, IBNTXA analgesia is restored. It does not restore morphine, and this is consistent with the role of 6-TM in IBNTXA, but not morphine analgesia. And this the next slide just gives you an overall summary of the pharmacology of morphine versus IBNTXA, which we believe is probably due to their relative selectivity, functionally at least, for the 7-TM and 6-TM receptors. This leads us to the question of how to examine the 3' splicing and the role of 3' splicing in opiate actions. To do this, we actually generated some additional mice, And we'll talk about the two of them, the E4M and the E7M. What we did here was we inserted a stop codon at the very beginning of exon 4 or at the very beginning of exon 7. What this does is it stops translation, but does not stop the actual expression of the receptor. So the receptor is still being made, but that you just lose, for exon 4, you just lose those 12 amino acids at the C-terminus. For the exon 7 mouse, what happens is you block only exon 7, so all the other splice variants are made under, normally. And when you take a look at this, you get a very interesting pharmacology. As you can see here with the exon 4 animal, so in other words, this is the loss of only the 12 amino acids on the exon 4 for those splice variants. You can see that wild-type animals for tolerance develop tolerance over 5 days, which is not unusual. The knockout animal, in other words, the animal that has the truncation of exon 4, develops tolerance more rapidly. But when you take a look at the animal where you have truncation of exon 7, you see the exact opposite. The wild type, again, develops tolerance over 5 days. But the animals with the truncation of exon 7 do not have any real tolerance development over this short period of time. So this would suggest that the exon 4 variants are important in maintaining opioid action. And it's interesting, since there's a recycling sequence in that 12 amino acids that we lost with this animal that restores the receptor back to the membrane. And the exon 7 appears to be important with regards to tolerance in the opposite way. Finally, if you take a look at CPP, you can see that the truncation has a significant drop in the liking score, the difference score for morphine, implying that exon 7 variants are important for reward behavior. How might we explore this further? To do this with the help of DiscoverX, we actually generated some clones where we took the various different splice variants that you see here and fused on their protag sequence to be able to look for beta-arrestin recruitment. If you take a look at the ones we chose, take a look at the variability in their amino acid sequence downstream from exon 3. Several things are notable. First, the actual length and the number of amino acids in the sequence. Second is the variability in terms of potential phosphorylation sites, particularly as you look at exon 7 variants, which are MOR1C and MOR1O. As you can see here, we've got quite a few serines, threonines, and even a tyrosine that are potentially phosphorylated in addition to the sites that are located within exon 3. So, how might these affect things? To do this, we actually used two approaches. We looked at GTP gamma S binding in a traditional binding assay. To assess the beta-arrestin recruitment, we used the Pathhunter functional assay. After cloning in the protag onto the receptor, DiscoverX was able to generate cell lines that were expressing these different variants with the ProLink tag. At that point, we went and made stables of these and utilized those to actually look at beta-arrestin recruitment. The beta-arrestin recruitment is a complementation assay in which the protag actually has a portion of beta-galactosidase and the beta-arrestin has a second portion. When they come together, you activate the enzyme and then you convert the substrate and get chemiluminescence. It has the ability to have high throughput, and we were able to do these reactions in 384 well plates very efficiently. We then calculated the bias by comparing the S35 GTP gamma S binding in the same receptor cells as we did the path hunter assay, so that the GTP gamma S binding is actually in the cells that have the receptors with a PK tag. When you put that PK tag on and you do a traditional receptor binding assay, what you can see, it has little effect on the actual affinity of the drug for the receptor, and this was very reassuring. It also has little effect in terms of the ability of the drugs to stimulate GTP gamma S. DAMGO has been widely used to assess opiate function in cell lines. It's a full agonist, and as you can see here, when we looked at these different variants, we were able to look at beta-arrestin recruitment, which is in blue, and GTP gamma S, which is in red. MOR1-PR is basically the MOR1 receptor in which we've truncated at the end of exon 3. So this has no substitutions at the end of exon 3. Then you have MOR1, MOR1A, B, C, O, and E. C and O are the ones that have exon 7. Several things become quite interesting here. First, you can see that if you take a look, the curves are relatively superimposed for O and for B1, but they seem to spread apart for MOR1C. And without getting too much detail of individual variants here, if you take a look at other drugs, you see other differences in patterns. So if you, again, you take a look here, you see that against the truncated form of the receptor, that the beta-arrestin gives you approximately a 35% stimulation relative to DAMGO, which was the standard. On the other hand, if you take a look at the maximal values of beta-arrestin recruitment for some of the others, you can see a dramatic decrease in beta-arrestin, especially for MOR1A and perhaps for MOR1C. Metenkephalin RHV is referred to as the heptapoptide. It's a peptide. It has high affinity for the mu receptors. And here some of the differences become even more dramatic. And whereas if you take a look at MOR1C, the curves for beta-arrestin and GTP gamma S are virtually superimposable. And yet you take a look at MOR10, and you can see a wide change. This is approximately three orders of magnitude difference in potency here. And you can also see very large differences in efficacy, in the sense that when you take a look at the truncated form, the RHV compounding is able to give you 100% of the expression relative to DAMGO, but if you take a look in E, it's only 50%. And similarly in MOR1 and MOR1A. So that what you have here is differences in potency and intrinsic activity at the different receptors for these drugs. And it's not clear that this is the same for every drug and every receptor. They seem to vary enormously. This is methadone, and it looks quite different from morphine. Again, you can see the superimposition of MOR1B and the truncated one. And look at the wide separation that you get with MOR10. Furthermore, if you look at the maximal effects of beta-arrestin against MOR10, it's really a very profound partial agonist with only a 50% response, whereas if you have the truncated form, it's actually more efficient than DAMGO. If you take a look here on B1, it's again fully efficacious compared to DAMGO. So what you have again is another example of selectivity in terms of that. Buprenorphine, I mentioned before, is particularly interesting because it's becoming more widely used clinically. And here we've observed a very interesting effect. We see differences for GTP gamma S, and you can see differences in efficacy between, for example, MOR1C and MOR1. So that does vary from variant to variant, but in no case were we able to see any recruitment of beta-arrestin by buprenorphine in any of the variants. Furthermore, if you included buprenorphine with DAMGO, it antagonized the effect. So the take-home message on this is that these different splice variants are functionally quite distinct and different. If you actually calculate a bias factor and you take a look at the patterns you can actually see this more graphically. By comparing comparing multiple drugs against a single variant, all normalized to DAMGO, what you can see is that a wide range of drugs that their bias factors differ enormously. That you go from a beta-arrestin bias to a G protein bias, or somewhere in the middle. The MOR1A shows this even more dramatically, where the orange feed compound is now very much G protein biased. Again, that extends to MOR1B1 and MOR1E, but not so much for the other two. Levorphanol was particularly of interest because of its extraordinary G protein bias at the MOR1O. You can also do a comparison where you actually look at a single drug across multiple splice variants. And here, again, you can see very dramatic differences. So everything is normalized to itself. And as you can see, as you go from MOR1 to MOR1O, for DAMGO, you see the appearance of a marked beta-arrestin bias. Levorphanol is the opposite direction. So again, there are multiple patterns. It's important to remember that when dealing with opiates and trying to understand their actions in vivo, that a single drug is going to be interacting with all those splice variants at the same time, that these drugs have similar affinities in the binding pocket. As you remember, all the 7 transmembrane domain receptors have identical binding pockets because they're encoded by exons 1, 2, and 3, which are maintained in all of those variants. But they do vary in terms of their transduction. One way to think about this is to consider a small little collection of instruments: saxophone, trumpet, violin, clarinet, and bass. And imagine them playing a very simple tune like Row, Row, Row Your Boat, but tell the trumpet and the violin to play very loudly and the others to play softly. Now tell the same musicians playing the same instruments and the same notes, just to alter the intensity of how they play, so now the trumpet and violin are playing softly. Is it the same tune? Yes, it is, but is it identical? No. There are subtle differences here, and this is the type of situation we encounter when we deal with drugs. So if you have a drug interacting with a multitude of different receptor subtypes, and the relative efficacy and potency varies among them for transduction pathways, you can expect to see similarities, but you may also see subtle differences. Remembering that when you give a drug, what you're looking at is the summation of actions of the drug against all the receptors at a single time. So in summary, I've presented an awful lot of material. I've shown you that the mu-opioid receptor undergoes extensive splicing. It generates 3 general classes of variants: 7 TMs, which are full length, 6 TMs, and single TMs. I'd point out that other GPCRs have very different structures. There are many that have 2 transmembrane domain receptors. There are others that have 4, 3, and 5. I would suggest, however, that the single TM variants are particularly intriguing because, indeed, they are present in a large number of GPCRs. I've showed you that all sets of the variants impact opioid pharmacology, and I've also shown you that among the full-length variants, both potency and efficacy in G protein activation as well as beta-arrestin recruitment vary from variant to variant. And this is manifest in changes in their bias signaling. I have to give credit to the people who actually did the work. Our group has actually been very fortunate to have 3 of us working together, myself, Dr. Pan, and Dr. Majumdar. Dr. Pan is a truly exceptional molecular biologist who actually cloned probably 90% of the mu-opioid receptor variants that have been reported in the literature. Dr. Majumdar is a medicinal chemist who made many of the compounds that I discussed using today, particularly IDNTXA. I've also been very fortunate to have support, specifically the National Institute on Drug Abuse, which has supported my laboratory for many, many years. I have additional support from the Peter F. McManus Trust and the Mayday Foundation. And I've also been very fortunate to have had assistance from the DiscoverX Foundation, and starting with Daniel Bassoni and the DiscoverX CAD team, who helped me with the generation of those different receptors for the Pathhunter assays. Thank you very much.
Thank you, Dr. Pasternak, for the presentation. I would like to remind our attendees today that if you have any questions for Dr. Pasternak, please submit them in the question and answer box on your screen. So let's look at the few questions that have come in. Dr. Pasternak, the first question is, the functional studies you presented were carried out on mouse receptors. Are there similar differences among the human variants?
That's actually a very good question, and one that comes up fairly frequently is how much can you extrapolate studies on these mouse proteins to the human situation? We've not looked at the β-arrestin recruitment. That would require generating all the cell lines that we just discussed. But we have, over the years, looked at GTP gamma S binding in these different human splice variants. There, what we do find is a very similar variability in terms of both potency and maximal effect of different opioids for these different splice variants. So based on that, my prediction would be that the humans will show, if not the exact same effect for each specific variant, the humans will show the same type of variability of response, both in the bias as well as in the potency.
Thank you. The next question is, how do these findings impact the use of computer modeling in the design of new drugs from the crystal structures?
We've seen a lot of talk these days about using crystal structures to design drugs, and there was a very intriguing paper that came out a few months ago where they actually predicted the structure of an opiate from the computer modeling and then went ahead, synthesized it, and showed that it had activity. And to me, that's an extraordinary step forward in understanding the pharmacology of these drugs. However, I would emphasize that there are limitations to this approach. The major one being that to do the crystallization, they've usually truncated both the C-terminus and the N-terminus. So all the differences that we are seeing in our functional assays with the GTP gamma S and with the β-arrestin really would not be seen in this type of modeling because those structures aren't there. We really don't know at the crystal level what the differences those sequences make in terms of the structure of the receptor. So I think that the receptors are interesting. I think that the crystal structures are proven extraordinarily valuable. But if I were doing drug development, they would only be a start, and I think that you really need to look at the functional assays and look at all the splice variants.
Great, thank you. The next question we have is, you discussed the significance of two of the sets of mu receptor splice variants, but did not mention further the single transmembrane domain ones. Are these relevant?
I did not discuss those very much, but the evidence is strong that they indeed are quite important. As with only a single transmembrane domain, you would not expect them to actually bind ligand, and in our hands we have not seen any evidence that they do. However, they do dimerize with the 7 transmembrane domain variants, and in doing so, they provide a chaperone-like effect. And they can actually increase rather dramatically the abundance of the 7-TM by diminishing its rate of degradation from the cell surface. So if you do pulse chase experiments, you can find, in work that Dr. Pan has published, you can find that the single TM slows the rate of degradation and almost doubles the expression level on cells. You add the fact that if you actually use an antisense paradigm where you can selectively downregulate the a single TM by targeting the splice junction between exons 1 and 4, for example, you can impact morphine analgesia, implying that these single TMs are important in the ultimate manifestation of morphine actions. It may simply be the fact that that when you downregulate the single TMs, you're downregulating the total abundance of the receptors. That's one possibility, but it does indicate that they seem to be important. And I would also mention that there are a lot of GPCRs that, at least theoretically, could be generating single TM variants.
Great, thank you. The next question is, could you comment on the pharmacological relevance of splice variant 11?
I'm sorry, splice, of the E11 splice variant? Yes, I think the splice, the exon 11 splice variants, the 6TM splice variants, are quite interesting. I think they're going to be quite important, but they're also quite enigmatic. We have information that is very solid, at least in my opinion, but we also have many questions that remain. We're not clear on exactly how it works. Now, if you look at the crystal structure of the receptor, you can remember that when I showed that to you, the transmembrane domain 1 was far removed from the binding pocket. So that in that situation, you might anticipate that removal of that single TM may not necessarily impact the binding pocket to an extraordinary degree. Of course, it will have an effect on the structure of the protein, so we really don't know. Attempts to measure the binding directly to the 6TM have not been successful, but that may in fact be due to several possibilities. One is that we may not be getting proper expression, that it may require certain chaperones and expression patterns in certain cells to be able to get it to where it's supposed to go and be active. The other possibility, which we're more inclined towards, is that they don't act by themselves, but rather in conjunction with association with other proteins, possibly even other 7 transmembrane domain GPCRs. We do believe that by targeting in the opioid system, by targeting with drugs, targeting those 6TM targets, you can get a very unique pharmacological profile, and you can obtain drugs with a far safer safety profile and actually a broader analgesic profile in terms of neuropathic and inflammatory pain.
Thank you. Next question is, dopamine receptor heterodimerization has been implicated in the very important aspect of opioid receptor-mediated analgesia tolerance. Do you expect heterodimerization to differ with the truncation mutants?
We've been able to actually show that the 6TM will dimerize with other 7TMs. So the answer there is yes, they can dimerize, but of course one has to always be careful because these are very artificial systems. We're putting these receptors into cells that don't normally express them, and we're expressing them at levels that are far greater than you might see in a normal neuron, for example. So the answer is maybe. We do know that they are capable of dimerization, but whether that happens in the native with native expression is not yet clear.
Okay, we have one other question. Opioid rotation is often used clinically to regain analgesic effectiveness. How do these observations fit into this approach?
Well, that's actually, I find that a very interesting question in the sense that if you go back to one of the earlier slides I showed you, if you remember that row your boat slide, where if you consider that the actions of a drug are really the summation of multiple receptors, being activated at the same time, it's easy to envision that with one drug, you're going to get a much greater activation of one subset of receptors. However, a second drug may not activate those subsets to the same degree and may be more dependent on a second subset of receptors. If you make the assumption that activation profiles are going to be proportional to the tolerance of that receptor or that receptor system, then you can see that perhaps by switching from the subset number 1 to subset number 2 receptors, that a drug may actually show incomplete cross-tolerance. And incomplete cross-tolerance is the basic fundamental reason why the opioid rotation system works. Opioid rotation, for those of you with, who don't deal with the clinical aspects, is where a patient is escalated on an opiate until side effects intervene, and then what happens is the clinician will actually rotate them to a different opiate and oftentimes find that the second opiate becomes effective at doses far lower than you might have anticipated based upon the relative potency in naive people. So it's an explanation, but certainly not something that's been actually proven.
Okay, we have time for one last question. Which animal model would be similar or more similar to the human system regarding the splicing pattern?
That's a very good one. We do see species differences. We see some species differences in terms of some of the sequences. The general patterns are the same. We have most of our information on the mouse, mainly because the mouse provides us a genetic background in which we can do the studies with the knockout models. Rat knockout models are beginning to become available, and hopefully we can get those in the near future. So at this point, it's a very good question, but I don't think we have a good answer. I can tell you that some assays translate more closely to humans than others. So, for example, some of the thermal assays, activity in the thermal assay usually implies activity in humans. However, some of the other assays, the translation may not be as good.
Great. Thank you, Dr. Pasternak. That's all the time we have for today. For those questions which weren't answered, we will be replying to them via email. We'd like to thank you all for joining our webinar today. Please stay tuned for our next webinar on April 5th on functionally selective GPCR agonists as drug targets for hypertension and heart failure. Thank you very much. Thank you, Dr. Pasternak, and thank you all. Have a great day.